Hi, my name is Liew. I'm a bioinformatician at the Bioinformatics Core at University of Nebraska-Lincoln. Today, I'm gonna talk about Porcine Reproductive and Respiratory Syndrome virus, PRRS infection, and how it upregulates negative immune regulators and T-cell exhaustion markers. This work was published last year in the Journal of Virology, and the paper can be accessed at, with the QR code on the right-hand side. The virus is an envelope positive sense and single-stranded RNA virus. It belongs to the order Nidovirus, the family Arterioviridae, and the genus Porcine and it has a genome of approximately 15 kilobases. PRRS is highly host and tissue restricted, and it only infects pig. The primary target cell of the virus is the alveolar macrophage of the pig, short for PAMP. Previous studies on PRRS, especially PRRS-infected PAMPs, were cultured ex vivo and included bulk sequencing of PAMP cells. Since only a small percentage of those cells are infected with the virus, these studies were just getting the combined responses from both infected and bystander cells. Therefore, for our study, we took the approach of in vivo infection and also looking into the differential effect of the virus infection on infected and bystander cells. We also want to know the mechanism of how PRRS evades the host immune system. Our experiment started with 4-week-old piglets, which were infected with a recombinant PRRS strain that expressed GFP. At 7 days post-infection, PAMP cells were extracted, selected for CD163, the main cellular receptor for PRRS infection, and then sorted for GFP plus, the PRRS infected, and GFP minus, the bystanders, cells for RNA sequencing. The results of our study found that there were a lot more reads aligned to PRRS-infected cells, the GFP plus, compared to the bystander cells, the GFP minus. However, majority of the reads aligned to the 3' end of the viral genome, which encodes for structural genes. Nuclear factor kappa B is an important immune signaling pathway. We found that many of its inhibitors were upregulated, including NF-κBIA, NF-κBID, NF-κBIZ, and TNF-AIP3. The upregulation of these genes help prevent excessive inflammation and protect the lung from immune-mediated damage. Multiple genes involved in the induction of T-cell exhaustion were also upregulated. This includes PD-L1 and PD-L2, the two immune-suppressive transmembrane proteins that interact with the PD-1 receptor on T cells and drive T cells into a state of exhaustion. Also, I want to bring your attention to the enrichment and activation of T-cell exhaustion pathway in the differential pathway analysis of GFP minus versus GFP plus cells, the bystanders and PRRS-infected cells. In summary, there is a significant overlapping of gene expression profile of the bystander GFP minus and PRRS-infected GFP plus cells. The T-cell exhaustion and nuclear factor kappa B signaling pathways are activated in PRRS-infected GFP plus cells. This might lead to suboptimal adaptive immune responses, which then might help the virus establish persistent infection in the host animals. Our findings help us better understand the virus immunopathogenesis. And last but not least, I would like to acknowledge everybody on the list, especially the Agriculture and Food Research Initiative and the USDA, National Institute for Food and Agriculture, which funded the research. And that marks the end of my presentation. Thank you for listening. Please stop by my poster if you're attending the conference in person. Thank you.